Hey guys, welcome back to biology. Today we're going to be discussing translation, which is our final step in the Central Dogma, which we've been learning about for the last two classes. So before we get into things, I wanted to go over all of your materials like I always do. So you'll need your guided notes open. You're going to follow along in these as we go through the slides. I suggest putting the video and the notes split screen so you can take both, or if you want, you can handwrite these and upload an image. Either one works for me. Then you have your independent practice. So for your independent practice, you guys are going to read a story about a castle and then the story of transcription and translation, and you're going to make analogies between the two. And then finally, you're going to create a comic strip that shows us the central dogma. You can be as creative as you'd like, but you want to create an analogy that shows us that process of going from DNA to RNA to proteins. Your strip should have 4 boxes. You can handwrite this or you can draw on these using like some sort of software, whatever works best for you. And then your final assignment for today is your exit ticket. So remember, exit tickets, you shouldn't cheat and you shouldn't use your notes. You can do corrections later, and this helps me see what you've learned in class today. So let's go ahead and get into it. All right, so to get us started today, you guys actually have a catalyst. I know you guys have missed those. So go ahead and pause the video right now and then answer this at your own pace. Once you've got your answer, come back and we'll go over the answers together. All right, so here are our catalyst answers. You guys don't have to have these exactly, especially number 1. Number 2 does need to be exact. So for number 1, transcription is the way that RNA is made so that DNA can travel outside of the nucleus and be translated into proteins. Remember, the DNA is too big to fit out of the nuclear pores, so the RNA is a necessary intermediary for that. And then our answer for number 2 is GACUCAU. I noticed a few people had trouble with this in their independent practice. So during office hours on Tuesday, if you show up for the first 5 minutes of mine, we're going to do some practice with this just so you guys can get that skill down. All right, so let's get into it. Here's our agenda for today. You guys just did your catalyst. We're going to do notes and guided practice. I have a quizzes for you to take that you'll do on your own. It's not for a grade, it's just a completion credit. So try your best on it, but don't cheat. It's just a way to see what you learned. And then you'll have your exit ticket. We won't have a case study, so don't worry about that part. So here are our learning targets for today. You guys need to be able to explain the process of translation. Remember, we've already learned about replication and transcription. Translation is our final step. And identify how errors in translation lead to genetic disorders. Next class, we'll get into this one more specifically when we talk about mutations. So remember the central dogma we talked about before. If you're not familiar with the word dogma, dogma is an overarching belief. It's often used in religion, but sometimes in science as well. So it's an authoritative principle considered to be absolutely true, right? And our central dogma in biology says that information flows from DNA to RNA to proteins. This process was first defined by Francis Crick, and it's been proven ever since through experimentation. So remember, the central dogma has 3 steps. We've already learned about 2 of them. The first one, replication, takes DNA and makes copies of it. The second one, which is transcription, takes those copies and then creates RNA from them. Remember, RNA is just a usable version of the DNA, one that can escape the nucleus and reach the ribosomes to form proteins. So today we're focusing in on translation. It's the final step where we go from RNA to a usable protein for our body. So remember, to get from RNA to proteins, we need something to tell us what this means. It'd be like trying to speak to someone who only speaks Portuguese and you speak English. So what you need is a translator, right? And that's what this process is called, translation. So that's how I remember this step. This is just some quick review from last class. Remember, transcription is when we're converting DNA into RNA. And last class we talked about this, but this class you should get them down if you didn't last class. There are 3 types of RNA. All of them are integral in the translation process. So remember, it's a chain of nucleotides like DNA, except the difference is it has uracil, and it is our link between the DNA and the RNA. So there are three types. The messenger RNA is the one that copies the DNA and actually travels from the nucleus to the ribosome. So it is a messenger, it's delivering that message. Then we have the ribosomal RNA. It actually becomes a part of the ribosome. And then the transfer RNA, which actually helps to build the proteins during this process. So we're going to watch a brief preview video that'll just kind of walk you through the steps we're about to learn about.
Translation begins with the binding of a...
1 second. I'm going to go ahead and hide my face. You don't need to see me for this part.
Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid. Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the 3-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds.
Watch the video. If you didn't get everything, that's okay. You can go back and rewatch, or you can just pay attention during the notes because we're going to go into all the details they discussed. Before we get too far, remember your body can't function without proteins. So this goes all the way back to our biochemistry unit where we discussed the different macromolecules. Proteins form your tissue, enzymes, hormones, membranes. Most of your body is made out of proteins, along with other macromolecules. So these are integral to human function and just to life in general. Proteins are made out of amino acid chains, which we've learned about as well. And your body only makes 12 of your 20 different amino acids. So the other 8 you get from your diet. We'll learn more about those 20 different amino acids when we move into using a codon wheel later in this unit. Remember in the video they discussed ribosomes. Ribosomes is where proteins are made. So this is where translation happens. It's where proteins are synthesized. And remember, synthesis is just a fancy word for creation. So the messenger RNA, remember it's a messenger, so it's carrying the information from the nucleus to the ribosome during this process of translation. Translation, again, is when we take the RNA and turn it into a protein. It happens in the ribosome, and it's our final step of the central dogma. Remember, RNA and DNA language uses nucleotides. So for DNA, that's A, T, C, and G. For RNA, that's A, U, C, and G. But for proteins, they use a different language entirely. They use amino acids. Now, we're going to do a quick check to see if you remember what we've learned so far. What we're going to do, I'll post a question. You can pause the video. After you pause the video, you can play it to see if you have the correct answer. So, question 1. Which of these processes does not use DNA? Go ahead and pause. The correct answer is, of course, translation. So DNA replication just uses DNA. Transcription starts with DNA to give us RNA, and then translation uses the RNA to produce proteins. Question 2. Nucleotides make up two molecules that carry genetic information, blank and blank. Go ahead and pause. And of course, our correct answer is DNA and RNA. These are our molecules that can hold that genetic info. Amino acids make up blank. Pause. Now let's check your answer. The correct answer is proteins. Remember, amino acids are the monomers of the polymer protein, which we learned about in biochem. What molecule carries genetic information from the nucleus to the ribosomes for protein synthesis? Go ahead and pause. Remember, the molecule responsible for that is mRNA. It's our messenger, so it's delivering that message. Where does translation occur in the cell? Go ahead and pause. And the correct answer is the ribosome, which we've seen a few times during this presentation. What two processes in the central dogma come before translation? You can pause. The correct answers are replication and transcription. So let's move on to the final part of our notes. We're going to go into detail on this process of translation. So remember, translation is how a protein gets made. There are a couple steps. Step 1 is that after transcription, our messenger RNA leaves the nucleus with our genetic information. Step 2, the mRNA actually makes it to the ribosome. So this is when all of the different types of RNA work together to produce a protein. So the mRNA comes to the ribosome here, that's this blue part, and inside the ribosome is the rRNA, or the ribosomal RNA. Together with tRNA, the mRNA produces a protein. Now, tRNA carries the actual amino acids, which we can see depicted here as the circles. The way I remember that is that the tRNA is transferring the amino acids. So it's carrying them here and then dropping them off to form the protein. And then step 3, the ribosome and the tRNA actually build that protein by connecting different amino acids based on the bases here on the mRNA. So they read the bases, figure out which amino acids are needed, and then form a protein. It's the number and type of these amino acids in the order that determines the protein that's produced. So that's the end of our notes. You guys are going to do a quick quizzes just to see what you learned from the notes and if there's anything we need to review before next class. After that, remember, you're going to move into your independent practice right here. So for your independent practice, you're reading these 2 stories, answering these 6 questions, and then creating your own comic book on translation. It only has to have 4 panels. And you can be as creative as you'd like, but remember, you're showing us how we get from DNA to RNA to protein. It doesn't have to be a literal image. You can use whatever format you want. And then to end out, you're doing your exit ticket here, which I will then use to assess what you guys learned and what we need to review. Remember, you shouldn't cheat, you shouldn't use your notes, and you can always do corrections later. Thanks again for popping into biology. Let me know if you have any questions. You can come to office hours between 2:45 p.m. and 3:45 p.m. And remember, my class hours are technically between 9 and 11 a.m. So if you review this before then and just have a quick question, you can comment on Classroom, send me a text, email, or call me. If you have kind of detailed questions, just pop into office hours and we can get those answered for you. Thanks.